And with Mike Young, showed that her and another protein, timeless or TIM, that together they molecularly respond to light through cycles of degradation, and that this is the core of the clock. Mike later showed that cryptochromes are also important for circadian rhythms of a different sort, and really has been at the heart of driving the field forward. But I just want to draw a slight contrast. So I mentioned yesterday that Eric Lieshaus really comes or represents part of an arc of fly geneticists, and sort of really, it's really a beautiful part of that continuation through developmental biology and using genetics to take apart the embryo. I would say that Michael really comes from a different strand that also had a not as long arc in Drosophila, which is molecular biologists that came into the field looking at the genetics that were emerging in Drosophila. So Mike himself was an RNA expert. I would include others in this group, Dave Hogness, Jerry Rubin, Welcome Bender, et cetera. And the analysis of phenotypes could be very powerful. And indeed, this was how the circadian clock was taken apart. So for this work in circadian rhythms, Mike became a member of Howard Hughes Medical Institute in 1989. He was elected to the US Academy of Sciences in 2003 and shared the Nobel Prize in 2017 with Jeff Hall and Mike Young. And as an aside, I would say it was about time. I think we were all kind of waiting for this. And so it finally dropped. With Jeff Hall and Mike Young for, quote, their discoveries of molecular mechanisms controlling the circadian rhythm. Really one of the fundamental discoveries in biology. And it's really exciting to have Mike here to talk about these. I have had the privilege to know Mike for a little while, a long while. He's a very thoughtful person. I learn something every time I talk with him. And he's really been able to walk across many disciplines and master them, which I'm very impressed by. He's been a strong voice for considering the impact of his and related work, circadian work, on the modern society, the disruption of our rhythms, and how that can affect our daily lives in very sort of fundamental ways. So for all these reasons, Mike, we're really excited to have you here. Thank you for joining us. Look forward to hearing about your work.
Well, thank you very much, Ross and Isabel and the people I know there. And it's a great pleasure to be here. I really appreciate the invitation, and I hope I can make a coherent presentation here. Let me share my screen. If I let's see what happens here. Slideshow. Let's see. Play from current slide. Is that visible and good? We're good.
Yeah.
Okay. Okay. So and then I'm going to also start my stopwatch here, so I keep on time. Here we are. The title is self-explanatory. I do hope I get to visit sometime in person. And so let me tell you and briefly summarize what I hope to do here. I'm going to talk a little bit about some personal history, introduce circadian rhythms, perhaps a little more briefly than I would have because Ross covers some territory there. I'm going to describe some contemporary work from my lab so you get a flavor of a seminar, not unlike what I would just give as a normal seminar. A couple of words about future challenges for the circadian field, because I think that's of interest for people, especially young people who are thinking about their future, and a few closing personal thoughts. Okay, so this journey that I've been in, this is a quote I often like to use. And so I'm going to start at the beginning and I'll stop when I come to the end, unless it's I'm grossly over time, at which point I'll stop a little earlier. So this story of mine began, of course, a long time ago, and I'm going to begin with a slide that even precedes my appearance. And I've shown this a few times just to indicate that, we're all immigrants in one way or the other. This is my father's, from my father's passport, my German passport. Actually, it wasn't a passport, it was a Fremdenpass. It was what the German government in the 30s exchanged for passports, a stateless UN document for Jews in Germany. And this was his immigration visa to the United States 6 years before I was born there. And so, whoops, there we go. So let me sort of walk you very briefly through a couple of salient events because they may be of interest. So let me say something about childhood to lay the foundation for what I called my somewhat uneven journey. And I think you might find this of interest. I've actually published this piece of data at some point in the last few years. But this is my report card from first grade when I was 5 or 6 years old. And I want you to pay attention to the right-hand column, the unsatisfactory there. And you see shows self-control, unsatisfactory, work habits, follows directions, unsatisfactory, works independent, is neat and orderly, unsatisfactory, unsatisfactory. So the point of this data is that I think a certain phenotype was set very early before other influences could be attributed. So I'm a fan of hardwiring, genetic influences, and so as I'll dictate a little bit later. And so this just sets the stage, if you will. I want to say a word about the two places I went to university, and simply say that I owe a great piece of my career to those places, not only for the education I had and for the people I met, but for the fact that those two institutions are extremely permissive in terms of misbehavior or lack of adherence to social norms. They're famous for having weird students. And that was very fortunate for me. I think if I had been raised in Western Europe or gone to university in Western Europe, I would have never ended up doing much in life because I was just too unruly. And in those two places, they were very willing to tolerate deviant behavior. So I'm very fortunate from that point of view. Let me say a word about coming to Brandeis. As Ross mentioned, I came here 47 years ago and took an assistant professor job. And I'm a great believer in not only genetics, but in the accidents of life, happenstance. And so Jeff Hall, who's on the right there, arrived at Brandeis 6 months before I did. We're almost the same age. We were in the same department and we became fast friends. And that friendship proceeded by 8 years. I would say any collaboration on our part. This is a picture from those early days, listening to a seminar in my house, somebody speaking in the evening, and we were both engaged in two of our favorite activities, stimulants, alcohol and tobacco, which fortunately, the tobacco, at least I gave up a very long time ago. So anyway, and it's really from Jeff that I learned about fruit flies and circadian rhythms, because as Ross mentioned, the Seymour Benzer was really instrumental. I'll come to that in a moment. And Jeff was a postdoc of Seymour's, and so he knew about this very well. So let me begin by introducing the topic of circadian rhythms. The terminology comes from the Latin circa dia, which means about a day, about 24 hours. Look at my hand here, about 24 hours. And that's because the circadian periods are not precisely 24 hours. They're a little shorter or a little longer than 24 hours, depending on the species. And it's really due to the light cycle, the light-dark cycle, which is, of course, precisely 24 hours, which resets the clock every day and resets, in the case of humans, our slightly long period clock of 24.5 hours back to 24 hours. And that's how come we are faithful every day to the day before, except, of course, when we do silly things like travel internationally and so forth. So the purpose of rhythms is to anticipate external events. And the early bird gets the worm, and the early worm avoids being eaten by the bird. And so that's the major purpose of circadian biology, namely for the three great principles of biology: to find food, to find mates, and to avoid predators, all of which involves being aware of what's happening in the outside world. And there's also a contribution to what's commonly called internal coherence. That is, biochemical events inside the organism are often spaced temporally, or to guess at what Eric Wieschaus might have talked about, spatial separation is one way to keep things organized, and the other way to do it is to separate them temporally. And that's what happens inside many animals. And so circadian rhythms arose long ago as adaptations to the rotation of the Earth, this inexorable light-dark cycle, and they're nearly ubiquitous. And the animal, I think, the organism which best captures this is cyanobacteria. Cyanobacteria have a very, very well-studied circadian clock that is the subject of exploration in many labs around the world. And cyanobacteria is responsible for oxygenation of the atmosphere. That is the ancestor of cyanobacteria, probably something very similar to what cyanobacteria are today. And so this photosynthetic, we owe contemporary life to this photosynthetic circadian organism. And to move to animals, that I'll simply summarize a circadian and actually genetic phenomenology by saying that locomotor activity, that is the running or the movement of animals as a function of time of day, is a fantastic assay. And that is because single animals can be very faithfully recorded. For instance, in this case, the activity of these single animals as a function of time of day in constant darkness, and here a week or 9 days of activity. And what you'll notice is that the activity begins and ends every day a little bit earlier, a little bit earlier than the day before. And that indicates that the free running period of this hamster is less than 24 hours. But if that animal is in a light dark cycle, then because of the entrainment, because of the locking on to the light dark cycle, the activity is exactly coincident with the 24-hour period. And you'll notice this animal is also nocturnal, very common for experimental rodents. And so since we love genetics, I want to simply say that this phenotypic assay, the fact that single animals in fruit fly as well as rodents can be assayed extremely faithfully, this is a remarkable phenotype. And it's not the case for most behaviors that individual animals can be faithfully recorded. So the mantra for genetics, like the mantra for real estate, which is location, 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 the mantra for genetics, in my opinion, is phenotype, phenotype, phenotype. And so Drosophila locomotor activity rhythms are assayed in this classic system. that was used by Konopka and Benzer as well, a single fruit fly in a tube, and then food at the bottom, a stopper at the top, and the animal runs back and forth. And it's this assay that was used in this fabulous paper by Konopka and Benzer, in which they isolate these three alleles of the period gene, an arrhythmic mutant, a short period mutant, and a long period mutant, all of which were allelic and single point mutations from this gene period. And most people don't realize that Konopka brought this problem to Benzer as a 19-year-old graduate student. He knew about rhythms from his collecting butterflies as an undergraduate, and went to Seymour and said, I think this would be a wonderful problem to explore with single mutant genetics in fruit flies. And so, as Ross mentioned, it was really Paul Harden in my laboratory who discovered that the messenger RNA from the period gene underwent circadian oscillations, and these oscillations were coincident with the behavioral oscillations that had been previously observed. And the distance between RNA peaks for the per short genotype was 20 hours, whereas the normal distance between peaks was 24 hours. And that's exactly what happens with the behavior. So we proposed that there was a feedback loop in which the period gene and subsequently the period and timeless proteins, as Ross mentioned, fed back and inhibited their own transcription. And of course, we subsequently learned about the proteins which drive the positive transcription of these negative regulators. And it turns out, so this is just a cartoon of this feedback loop in which two proteins, the cycling of two proteins are indicated, and they turn out to be landmarks for us. The clock transcription factor, its mRNA, and the period or timeless mRNAs. And the reason I'm showing these two transcripts, the clock gene's transcription and the period gene's transcription, is because they'll be important for something that I'm going to say a little bit later. And so it turns out that this oscillating gene expression at both the RNA and the protein level segues to the fly brain, and in particular, the approximately 75 pairs of circadian neurons, which are present on each side of the fly brain. And they're stained here with an antibody against the period protein in red. And there's also a neuropeptide called PDF, which is stained in green, a surrogate for this neuropeptide. But the main point is that there are only, by immunohistochemical assays, there are only about 150 neurons out of the 100,000 central brain neurons in the fly brain, which are easily, in which clock gene expression is easily visible. So it would seem that these 75 pairs of neurons are prominent and important. And of course, they represent only about 1 in 1,000 of the total neurons of the fly brain. And importantly, there has been over the last 15 years or so important connections made between subsets of these 150 neurons, 75 pairs, 75 per hemisphere, and the normal locomotor activity pattern that one sees from fruit flies. And this characteristic activity pattern, namely a peak of activity in the morning, a siesta in the middle of the day, a peak of activity in the evening, and then quiescence, in fact, sleep at night, is characteristic of many, many insects, this crepuscular or dawn and dusk bimodal pattern. And we know and love this pattern from mosquitoes because, of course, they're very active at bedtime and in the morning and much less active in the middle of the day and the middle of the night. And specific subsets of these neurons have been connected to particular features of this activity pattern. For instance, these cells here to morning activity, these cells here to the siesta or quiescence in the middle of the day, and these cells here have been connected to evening activity. So this has been a rich source of information where we could connect particular subsets of circadian neurons to particular features of the behavioral paradigm. So we were interested in delving more deeply into the molecular features of these circadian neurons. How heterogeneous are they, and what do they express? And we took advantage of a driver line that uses a piece of the clock promoter to explore in more detail the molecular heterogeneity of these circadian neurons. And this driver expresses in most of these 150 neurons with only a few ectopic cells and a few cells missing. I can't quite, let me see if I can change this view. Here, I'm afraid to mess around. In any case, most of these neurons are expressed very handsomely, and there's no glial expression, most importantly, because glia also have prominent circadian clocks. And so we took this system and we purified these neurons by fact sorting, and we collected collected flies around the clock, because of course we want to look at gene expression as a function of time of day, and then fact sorted these neurons and prepared sequencing libraries so that we could do single cell sequencing and examine these neurons in detail. And this work was really spearheaded by a postdoc in my lab, Dylan Ma. And of course, it's thanks to Dylan and the rest of the lab that I have something to say here, and I still am excited about work going on in the lab. And so it turned out that when these data were analyzed, the single cell sequencing around the clock, there was really striking heterogeneity from these 75 pairs of neurons. And in fact, they distributed into a large number of subclusters, shown here on a Tisney plot, and that the marker gene expression was remarkably discrete. So here are these 39 clusters, and these are the top 5 marker genes. And what you can see from the heat map is that there's really discrete expression in all these 39 clusters. And the reason there's a subset of them numbered and colored is because I'm going to focus on these 17 colored clusters, because they are what we call the high value clusters, where we could connect individual clusters here with individual anatomically distinct circadian neurons. And what I want to point out to you is that for all of these 17 clusters, the cycling of the timeless gene in each of these clusters is quite similar, with the peak in the middle of the day. These are 6 time points, the 6 time points we collected and purified. And for each of these 17 clusters, the timeless messenger RNA has a profile which is very similar, not identical, as you can see in all cases, but very similar, with a broad peak in the middle of the day. And what's shown in blue are the data from a light dark cycle that is in trained animals. And what's shown in gray are the data from animals collected in constant darkness after 3 days of constant darkness. And what you can see is that the endogenous program without any light dark cycle is fairly similar to the program that we see in a light dark cycle, not identical, but really quite similar. And that when we look at clock gene expression from the clock transcription factor now, what you can see is what I alluded to in that cartoon at the beginning, that the profile or the phase of clock expression, just focusing for the sake of simplicity here on this single cluster number 2 here at the beginning, you'll notice that this has a trough in the middle of the day, whereas timeless has a peak in the middle of the day. And that's characteristic of most of the clusters. Here's another one that's very clean, this cluster number 1 here, and here's the timeless expression, and here's the clock expression. So the head RNA that had been analyzed years and years ago mostly comes from eye photoreceptor cells and other cells. But these 150 neurons, which are really invisible in head RNA, have a cycling pattern, which is very similar to what's seen in other tissues. And so it turns out that marker genes, and to some extent knock-in genes, which I'll talk about, which I'll show an example of in a couple of minutes, they turn out to be very useful to be able to superimpose these single cell cluster data onto the anatomical pattern here. So these two clusters, 25 and 2 here and here, they turn out to map. I can't see this pattern over here, but they map onto the small LNVs, and that's because these express, see if I can go backwards, these express the neuropeptide PDF, and it's the only place that the neuropeptide PDF is found, and that turns out to be characteristic of the small and large LNVs. PDF is only here. And what you can see in this animation is that different neuropeptides identify specific subsets of these circadian neuron clusters. And we know from staining with antibodies against those neuropeptides that where on this anatomical map those neuropeptides show up. And so one of these neuropeptides, trypsin, it turns out that that a lab in China made a knock-in into the trypsin neuropeptide gene, and that knock-in expressing GFP in the natural gene only expresses in 2 cells, and those 2 cells are a subset of the 4 LND cells identified by a timeless antibody shown here on the merge, and that tells us that those 2 red cells right there represent this cluster from the single cell sequencing. So this general approach has been extremely useful. Has been extremely useful. And just to elaborate a bit on the general expression patterns, these are 17 clusters, and the heat map for the top 3 markers. These turn out, this particular heat map identifies transcription factors, which are present and specific to these 17 clusters. And you'll notice, for instance, in this subset of clusters, so-called DN1, mostly DN1 clusters, there turn out to be remarkable specificity to the transcription factor expression, something we're currently working on. And GPCRs also turn out to be very beautifully specific to individual cells and clusters, something that we're exploring from a network standpoint. And here's a particular GPCR that we're interested in for behavioral reasons, including the regulation of sleep. And what you'll notice is this GPCR is not only expressed in specific clusters, but it undergoes very prominent circadian cycling in subsets of these DN1 clusters. So the guess is, of course, that gene expression, something we're trying to validate, that gene expression regulation is contributing to the sensitivity to dopamine in particular clusters, particular regions of the circadian network, and then passing that information along to the cells to which these particular circadian neurons are connected. And it turns out that the cycling of particular genes, transcription factors here, also turns out to be, interesting and important. And so a general question I want to raise for you is this striking clock neuron heterogeneity, that is the fact that we have perhaps an average of 2 to 3 cells per cluster, or 2 to 3 cells for a specific cell type, is that unique to the circadian system? And I want to suggest to you that the answer is no. And of course, this is work in progress. But let me first point out that Li-Ching Lo's lab has done single cell sequencing of projection neurons from the olfactory system. And the heterogeneity that he has described, about 3 cells per group or subcluster, is quite comparable to what we see in the circadian system. And of course, in his case, this fits with anatomical data as well, because the olfactory system is so well characterized. And so we've looked at dopaminergic neurons around the clock to ask the question, how heterogeneous are they? And our data indicate that there's comparable complexity in the central brain dopaminergic system. And without going into any detail whatsoever, I'll simply tell you that we find on the order of 20 or 25 clusters from the dopaminergic system, and the number of cells that are dopaminergic in the fly brain is not unlike the 150 neurons that we find constitute the circadian system. So once again, one finds a small number of neurons that constitute each cluster or each specific class of dopaminergic neurons. And so from these 3 examples, namely the clock's circadian system, the projection neuron pattern from Li-Ching Lo, and our preliminary data from the dopaminergic system, the suggestion is this may be quite general. And so, to take a 35,000 foot view, I wonder what pilots say in the metric system. Do they use feet, or do they say 10,000 meters? Anyway, to take a broad view of this system, I'll point out that the fly brain, to scale here, compared to the human brain, has about 1 million times fewer neurons, or even 1,000 times fewer neurons than the mouse brain. And yet, flies have this remarkable sophisticated behavioral repertoire that they can undergo. I mean, they learn memory, aggression, motivation, mating and courtship. You people know about all this. And so I think the question is, how does an animal with such a tiny brain undergo such sophisticated behavior? And I think it's fair to suggest that this is due to the fact that the brain is much more heterogeneous than what happens in mammals, so that circuitry can be generated with only 1 or 2 or 3 neurons in each element of a circuit. So the single neurons do the job of what would be 100 or 1,000 neurons in a mammalian brain. And so this is the answer to how the behavior can be so sophisticated. So let me turn and address the question of whether there are molecular clocks in the fly brain beyond the 150 circadian neurons that one can see by immunohistochemistry and that we characterize by single cell sequencing. And as Ross mentioned, the molecular clock is supposed to be ubiquitous. And certainly from mammalian data, the idea is that the clock is present all over the organism, not only in the brain, of course, but in spleen, liver, lung, skin, really everywhere. And we know, for example, that the suprachiasmatic nucleus in mammals is a circadian oscillator in vitro as well as in vitro. For instance, in hypothalamic slices, it undergoes all by itself a circadian rhythm of firing rate. And so in the brain, in the mammalian brain, the suprachiasmatic nucleus is not unique. It's been shown for the pineal and the pituitary that if you put them in vitro, they undergo circadian oscillations all by themselves, damping in the case of the pineal, but the point is nonetheless robust circadian oscillations. So the general idea is that also for the brain, there are lots, if not many, most places in which circadian cycling oscillations take place. And so although the SCN orchestrates and keeps in sync peripheral tissues oscillators, we think of many or most places as having a clock. And so if we turn to this question in the fly brain, we can ask the question, do we find clock gene expression, which is of course a surrogate for a clock from our point of view? Do we find clock genes elsewhere? And if we now use single cell sequencing methods and look for expression, in this case, for instance, of the clock transcription factor itself, and we look in the clusters from TH neurons, these are the neuron clusters, and they're actually co-clustered with the clock neuron clusters, we find only 2 clusters from the dopaminergic neurons. I should have said that TH, tyrosine hydroxylase, is a pseudonym for dopamine. We only find 2 clusters where we find credible expression of clock genes, shown here for clock, but this is also true for other clock gene expression, timeless, for instance. And so if we turn to another kind of assay, but where we ask the same kind of question, what happens to clock gene expression in dopaminergic neurons, TH neurons, as compared to our circadian neurons? And here at 2 times a day, and this is done now by bulk RNA sequencing, and this just gives you a flavor for the order of magnitude expression pattern for the clock gene at its peak and its trough at the beginning of the day and in the middle of the day from the circadian neurons. And here's the comparable data for the dopaminergic neurons. And what you can see is there's about a 20th as much expression from the dopaminergic neurons here, but still with a representative, or I should say characteristic pattern of more expression at the beginning of the day than at the end of the day. And this is more or less coherent with what one sees by analyzing the individual clusters. That is a little bit of expression, but much less than one sees from the clock neuron clusters. So with this as a benchmark or a proof of principle, one can now go to a third circadian driver, NSIB, in which, a third driver of gene expression, NSIB, which drives GFP expression in all brain neurons, and now ask the question, what do we see if we examine clock gene expression in all brain neurons? And the data are that we really only see background expression, barely any circadian gene expression at all. And no longer do we see more expression at the beginning of the day than the beginning of the night. So the title of the slide and our working hypothesis is that there's really vanishingly small expression of the clock system in most of the fly brain. Considerable here, a tiny bit here, and almost nothing when one scans the entire brain. And so we think that there are relatively few places where the clock is really expressed in the fly brain. And actually, in case I forget to mention it, we think that casts some doubt on the fact, at least for the brain, that the clock system is universal. And I think that also requires some re-exploration in the mammalian brain, which we're also trying to begin to look at. So I think the jury is out, especially for neurons in the brain, whether the clock is really ubiquitous or not. And I think the explanation for this may go back almost 20 years to a paper that Ravi Alada began in my lab and finished in his own lab when he moved to Northwestern, that suggested that the gene clock may be a master control gene like PAC6 for eye development, because when the clock gene was misexpressed, then it turned out that we could generate ectopic circadian clocks broadly in the fly brain with actually unusual behavioral patterns as well as molecular patterns. So it may turn out that the rules of where are clocks in the brain and how do they get to be regulated may turn out to lie in the regulation of this upstream transcription factor clock. And that's something that we're interested in exploring. How is that gene actually regulated in the brain? So what I've told you is that Drosophila-Circadian neurons are very heterogeneous. This may be a general feature of Drosophila central brain neurons and contribute to the economical use of these neurons for behavior. The clock may be present in only a small fraction of the fly brain. And so is the mammalian clock really everywhere? What about the cortex of the mammalian brain as a sort of future question of interest? So let me raise for you 3 challenges in circadian biology for the future, things that I think the field does not know. First of all, what really accounts for the timing? Why 24 hours? What are the rate limiting steps, to use a biochemical turn of phrase? And the reason for posing this question is simply it rephrases the fact that we have a broad sketch of how this works, that what we uncovered, this feedback loop, gives you a feature for how it works. And there are other genes that we know about that contribute to post-transcriptional regulation, turnover of the proteins, as Ross mentioned. And we know quite a bit about this, but we don't actually know what the rate limiting steps are. Why 24.5 hours in mammals? Why 23.5 hours in rodents? Why not 22 hours? Why not 27 hours? What exactly are the rate limiting steps? And of course, to really answer this biochemically specific question, one needs real biochemistry, and there isn't much biochemistry of animal clocks, a little bit, but not really very much. The second challenge for the future, I think, is leveraging what we know to improve human health. And sometimes I would spend 10 minutes on this topic, but I'm not going to do that today in the interest of time, but simply say that this list here encompasses, I think, the major features of human biology that circadian biology impinges upon. And I think it's very interesting to imagine the extent to which what we're learning in the field can contribute to these various aspects of human health. And the third general feature for the future is to understand the basic science of sleep. And so sleep is a conserved feature in the animal world. Even some single cell animals undergo sleep or a surrogate of sleep. And we know that fruit flies sleep, and they do so in ways that are extremely reminiscent of, if not parallel to, the ways that mammals sleep. Even aspects of the pharmacology are identical. And so I think it's likely, and I hope, that studying the fruit fly will unlock some of the mysteries of sleep. What is the deep conserved function of sleep? And if you will parallel, I think, the contribution that circadian rhythms, fruit fly circadian rhythms made to the general circadian field. So I would encourage anybody young out there interested in circadian biology to think about these three challenges and see if it interests them. And I neglected to mention in this context, darn it, that I think the big sleep question is, how does sleep homeostasis work? So in other words, we know about the circadian control of sleep, that it's very regular, the very regular contribution of the circadian clock is making us tired and alert at specific times of day. But who's keeping track of sleep need, how much sleep we have had and how much sleep we need, and where is that measurement device and what does it consist of, both from a neuronal standpoint and from a biochemical standpoint? Okay, so a couple of closing thoughts, again, specifically for young people. I think I get asked the question often, how could we do something great? How can we follow in the footsteps of the pioneers, the people who have made great contributions to a particular field, for instance, in Drosophila or circadian biology? And my advice to young people is to not confuse the means and ends, to focus on the means and not the ends. I think the ends, that is what will happen from your work, is hard to predict. It takes a lot of luck, and it's just not guaranteed. And the best one can do in life is to do the right thing, focus on the means, work hard, enjoy your work, most importantly, and let the future unfold as best as it can. And lastly, I want to have some sort of philosophical thought on genes and chance and destiny, a bit along the lines of how I began. I like this quote from Albert Camus: all great deeds and thoughts have a ridiculous beginning. It's often great things start in the most haphazard and silly of ways. We get inspired by something by chance. I think we have to acknowledge that genes make an important contribution not only to the fruit flies and to the work we do, but also to human beings. They tilt the table. In other words, I think it's illusory to imagine that we really have a blank slate. And I think it's important to understand or to appreciate the fact that genetics, human genetics now, what we inherit it's really all by chance. And two anecdotes that are, I think, useful in this context. When I was criticizing one day somebody who inherited money and didn't earn it, my wife turned to me and she said, well, you didn't earn your intelligence either. And so I think the idea of recognizing that the good things that we've received in life should introduce a sense of humility into how we think about what we've achieved, and also tolerance for people who have perhaps received an inheritance or even a genetic dowry that's less rich than what we've received. I mean, I guess the other human anecdote was my daughter, when she was very young, said to me one day, Daddy, how come I was born in the United States in this family and not born starving in another part of the world? And so I think where we end up and how we begin should introduce a sense of humility and tolerance. And so, like this previous slide, I said, enjoy research, work hard, but success or failure is only marginally under your control. There's a lot of luck and chance which enters into what happens. And so mostly, don't be too hard on yourself if everything doesn't work out perfectly. Enjoy the ride. And so with that, I think I'll end. And thank you very much. And I don't know if there are questions that occur, but in any case, I'm happy to hang around for a few minutes.
Thank you very much, Michael. Sorry about my ghostly appearance, but I wanted to still have something in the background I'll tell you about before. But thank you very much for that amazing talk, and also for the kind words for the young people. And I truly share those thoughts. And there are a few questions, so we'll go through a couple of them. Michael, there is also the Slack platform. I don't know if you are familiar with that.
I'm familiar, but I didn't go on it, and so you're going to have to read it out to me, or somehow transmit it some other way.
Okay, we can do that. So let me start with the question. So the first question is by Bhavna Verma. Why period gene has been linked to a non-circadian function in male courtship song?
So, what a terrific question. Thank you very much. So it's terrific because it's incredibly important. And you're a scholar, whoever. Well, what's the name of the person who asked the question?
Bhavna. She's actually going to introduce the next couple of speakers, so you're going to know her personally. Bhavna.
Okay. Bhavna. You're a scholar, right? Thank you for that. That work is very old, but it's very important. And the short answer to the question is I don't know. And I think it remains a mystery because, as your question implied, first of all, I need to tell everybody that that work is very controversial because there's been subsequent work published where some of the observations have been questioned, whether it's really true. And the courtship song was a 1-minute rhythm and was claimed to be subject to control by the period gene. And of course, it's very hard to imagine how a transcriptional foundation can regulate a 1-minute rhythm. And so I think the answer is, I don't know if it's true. There's a dimension which we don't understand. And of course, the other possibility, as underscored by the controversy, the experimental controversy, is that it's simply not true.
Great. Thank you very much. I think I can ask one more question before we move on. And what I will do after that, Michael, is going to text you in the chat, directly to you, the other questions. You can reply them to me, and I will put all of that in the Slack for people to look at, okay?
Okay.
But the next question is, were you able to look at the effect of sex and age in your RNA-seq data? Your single cell RNA-seq data?
So the answer is no. We didn't look at sex and age. Sex we should be able to simply pull out because we do have gene expression markers that should indicate male cells and female cells. And so we should do that. We haven't done it. I'm going to mention it today when I go into the lab later. And age we also haven't done. We simply haven't done it. We do have a project on aging, but you know, now let me just think if yes, we do have those data. I take it back. But I can't tell you what the answer is. What are the big changes from a circadian standpoint?
Okay. And maybe we can close with a general question. So the previous question was by Hamed Batmos, and he has a general question to you, and we could use that to close this session, which is what has been your greatest challenge in working with Drosophila?
What has been my greatest challenge? I think the honest answer is simply, I have to struggle to think of one. Of course, there's mostly advantages. I think the amount of material. So for somebody interested in biochemistry, which I am, mechanism, that getting enough material for certain kinds of experiments is a challenge. The people who work with liver, rodent liver, have buckets of material compared to stuff that we can get from the fly brain. So for certain things, flies are great, and for biochemistry, not so great.
Yeah, I agree, as I've tried a bit of the biochemistry as well with the fly. Great. I think I'll do what I said, that I put directly to you the other question. You can answer there, and I'll put that in Slack. People carry on writing also on the Slack, and we can make sure that reaches Michael in other ways, maybe in the chat.
I'll go. I have the Slack. Can you send me the Slack instructions, and I will log on to Slack in an hour or two and
Perfect.
And answer them. I'll answer them in Slack, and they'll all be in one place.
Perfect. Great. Thank you very much.
Oh, thank you very much.
And otherwise, we have Bhavna, as I said, to carry on with the next speaker. Thank you very much, Michael, and thank you, Bhavna.
Please.
Good afternoon, everyone. My name is Bhavna Verma. I'm currently working with Dr. Peter Lawrence at the Department of Zoology